Hi, my name is Michael Willis. I am an undergraduate working in Dr. Jonathan Busada's lab. This research was supported by West Virginia University startup funds from the National Institute of General Medical Sciences. My research this summer has been to develop an organoid system in vitro that will allow for research and future studies on the gastric epithelium and the effects of Helicobacter pylori. This research is relevant because gastric cancer is the fifth most common cancer in the world and the third leading cause in cancer death. Helicobacter pylori infection is linked to 90% of all gastric cancer cases and infects 50% of the world's population. The understanding of how Helicobacter pylori causes cancer and the treatment options are limited, this being due to the harsh environment of the inner stomach mucosa. To better understand Helicobacter pylori and its effects on the stomach, organoids can be microinjected with the bacteria. Our goal is to culture these gastric specialized organoids, which are three-dimensional organoids. They are stomach glands from mice and or humans, suspended in matrigel to form mini stomach systems. The controlled environment of the organoid culture allows researchers to ask questions that would be too difficult to ask in an in vivo study. These difficult to ask questions may be important to determine a model of how Helicobacter pylori infection causes stomach inflammation and develops into cancer. Down the road, organoids could be used to perform studies using patient tissue cultures. These could be patients that have had cancer caused from Helicobacter pylori infection. We could use possibly these patient tissue cultures to determine what's different about their stomach mucosa that allows for Helicobacter infection to cause gastric cancer or an inflammation. So to culture these organoids, a specialized cell line called LWRN, which produces proteins, Wnt3A, R-spondin 3, and noggin. And what they do is they condition media and allow these proteins to be suspended throughout the media, which will then go on to be important in the differentiation of stem cells into gastric organoids. Stem cells are introduced into the media where these key proteins supplied by the LWRN cell line should differentiate them into gastric organoids and gastric epithelial cells. The conditioning process involves growing the LWRN cell line to confluency in a flask and collecting and replacing the media on top of the cells in the flask every 24 hours, making one full cycle every 4 collections. After conditioning media, it is important to confirm Wnt activity. Go back. Shoot. Okay. So to determine Wnt activity, a top flash assay was used to confirm. And what it does is it's comparing the Wnt activity of each cycle to the activity of a 2x stock of commercial LWRN conditioned media bought from Millipore Sigma. The top flash assay involves another cell line called top flash cells, or HEC293. The top flash cells need to be plated in a 12-well plate and allowed to be grown to 80% confluency. When they're fed with the sample conditioned media, so cycle 1 conditioned media and cycle 2 conditioned media, and then also the commercial media. They're incubated for 10 hours and also 24 hours. And then the plate reader will then tell us the luminescence value or the Wnt activity of the samples. So you can see that the 2 cycles are about 50%, a little less. That's what we expected, considering we had a 2x stock of commercial LWRN. And so the activity was about half The true confirmation, though, comes from passaging organoids and determining whether or not organoids will grow with these media samples. So organoid media was made up with the samples and then passaged. And then here we see viable organoids from all the samples after 48 hours. After 48 hours after culturing. And so this means that the conditioned media protocol works and that the gastric organoids can be cultured using media that we can replicate in the lab. Next, the top flash assay will be used again. This time, we will be adding the third cycle. Also, we will be adding a well that has commercial media from the 2x stock diluted down to a 1x stock, and adding a purely negative control that will be just fed DMEM media. Organoids will also soon be passaged and cultured using all cycles of conditioned media. Future directions with the organoid project will involve microinjecting these 3D organoids with Helicobacter pylori to study its effects on the gastric epithelium in a controlled environment without any external factors, including immune functions. This will allow us to determine the model in which Helicobacter pylori causes gastric cancer. Using the conditioned media will be much more cost effective than buying the commercial conditioned media, and will allow our lab to further study organoids using our own resources for future projects. Just want to thank again Jonathan Busada, Dr. Busada, for allowing me to work in his lab, and also the institution for funding with the startup funds. Thank you.